Hi, you guys. Here's Carol, part 2 of chapter 13, where we're looking at clues to evolution, and we're going to look at fossils and anatomy and DNA. So the first one is fossilization and how this happens. So I gave you some examples here of how that occurs. It could be a mold or a cast. So a shell, for example, here, might have fallen into the mud, and then that becomes hardened, and what's left behind is the actual mold, which then fills in with something different, with dirt, and it becomes a cast of that actual mold. That's a possibility. This insect here has been preserved in amber. So you've probably seen that in jewelry when an insect has fallen into sap, tree sap, and it doesn't decompose because it's so thick and there's no air in there, and it'll be preserved. You could preserve in ice also. So organisms have different ways of getting sort of saved and preserved. Petrification here is when the actual cells that were there as part of the tree are no longer there, but other molecules fill in the spaces of what used to be a cell, and it ends up looking just like that structure, but the actual living part is no longer there. Number 4 here is an example of a compression where you just see sort of the impression of something that was pressed into mud that then became hardened. These are really fun to find if you're in a place where these fossils are prevalent. I think I told you my story of finding some trilobites. So these are some examples from your book that have, petrified wood. There are plants. Some of them are incredible, like, well, these are the trilobites that I used to hunt for, which is really fun. What's this one? This is an egg where you have an organism inside that's become fossilized. That's pretty cool. And this one here is pretty cool too. That's poop. Petrified poop is very interesting. It's got to be. And the ginkgo, it's an ancient leaf. Another interesting example is in Oklahoma, where it's full of marine fossils, and Oklahoma is nowhere near the ocean, yet these fossils are found up in the hills, indicating that that was ocean at some point in time. I've told you the fossil record is incomplete. Many organisms that have very soft bodies, for example, they never fossilize. Erosion sometimes destroys a lot of fossils when these rocks crack, so it's not like we have a complete fossil record. That would be so nice. So what we have to do is try and date some of the fossils that we do find. And we talked before about how the more superficial layers would have recent fossils and the deeper ones would have older fossils. But can you actually get dates on those? That becomes a little more complicated, but also interesting. So what we use to do that are carbon isotopes. Carbon-14. So let me tell you about that. And also I have a video posted if you need more on that by Mr. Anderson. So this is carbon-12, the carbon that you find mostly in nature. And it has 6 protons and it has 6 neutrons, and that's the carbon that we have been talking about when we talked about chemistry. However, in nature, just randomly, there also exists carbon-13. It's not very common. It's about 1.1% of the carbon found, and it has an extra neutron. And on top of that, we have carbon-14, which is even more uncommon, and that has 6 protons and 8 neutrons. So this makes it a very unstable molecule. So is this unstable. It's going to want to lose that neutron and become regular old carbon-12. So this carbon is what we can use to actually date fossils. How so? Well, think of this. if you're eating carbon, because you're eating these macromolecules, it is likely that you're eating a lot of that carbon-12 and at times some carbon-14, but a small amount of it. So in your bones, if someone were to go and look at your bones, you would have both carbon-12 and carbon-14 in your bones, and you'd have a tiny quantity of the 14. Okay? That's normal for everybody. Then let's say you die, and someone finds your bones years and years later, and they want to know how old it is. How can they tell? Well, carbon-14, I told you, is unstable, and over time it will convert, and it'll no longer be present as carbon-14. It leaves as nitrogen, but don't worry about that. Just, we go from having 3 balls out of this array of carbon when you're alive, and then when you're dead, you go losing it little by little. Now, if you knew how long it took to lose it, then you could date that particular bone, because that molecule is unstable, and over time it will vanish essentially from a dead bone. The only thing I want to show you that's new on this slide, it says decay, a neutron decays into a proton and it emits energy. It's actually radioactivity that's going on. It's radioactive and it loses that element. Isotopes decay. Radioactive decay is known as nuclear decay or radioactivity. Is the process by which a nucleus of an unstable atom loses energy by emitting neutrons and changing mass. So how do we use that? Well, if we knew how long it took to do that, we could date our bone. And we have learned that it takes approximately 5,730 years to lose half of that carbon-14 that was originally in the living bone. So this is a graph showing that while it's alive, it has all of the carbon-14. And then 1 half-life means 5,730 years later, it only has half of the carbon-14 it did originally. And then another 5,000 years later, 730 years later, it loses another half of that half, and so on and so forth. Losing. So if we analyzed that bone and saw here how much of that percent of that original amount of carbon-14 it had, we would be able to tell how old that bone was. That's how we use radioactive isotopes. We search for them, and when we don't find them, we know a certain amount of years have gone by. So the half-life is the term that we use. The half-life of carbon is 5,730 years. That's how long it takes to lose half of that carbon-14. I want you to watch that radiocarbon dating video from Bozeman Biology. That'll help you a lot. And I'm going to end there.